Good day everyone, this is Joecel, your assistant for today's video because we are going to explore another lesson, the history of microbiology. Microbiology is the study of living organisms of microscopic size. The term microbiology was given by French chemist Louis Pasteur. Microbiology is said to have its roots and the great expansion and developer of biological sciences that took place after 1850. The term microbe was first used by Sidelot in 1878. The history of microbiology can be classified into four different theories. They are Discovery Era, Transition Era, Golden Era, and Modern Era. Now let's discuss it one by one. First is Discovery Era. Aristotle proposed the spontaneous generation theory. He stated that living organisms could develop from non-living materials. In 13th century, Robert Bacon described that the disease is caused by a minute seed or germ. However, in 17th century, Robert Hooke, an English scientist, was the first to use a lens to observe the smallest unit of tissues he called cells. Soon after, the Dutch amateur biologist Anthony van Leeuwenhoek observed that what he called animalcules with the use of his homemade microscope. Leeuwenhoek was the first person to produce precise and core-text description of bacteria and protozoa using a microscope he made himself. Because of this extraordinary contribution to microbiology, he considered as the father of microbiology. Leeuwenhoek is also considered to be the father of bacteriology and protozoology. He wrote over 2,000 letters which were transmitted as a series of letters from 1678 to 1723 to Royal Society in London during a 70 years period. Next is the Transition Era, where microorganisms were known exist. Most scientists observe that such simple life form can surely arise through a spontaneous generation. That is to say, life was thought to spring continuously from mud and lakes or anywhere with sufficient nutrients. This concept was so compelling that it persisted until late into 19th century. The main aspects were to solve the controversy over spontaneous generation, which include experimentations mainly of Francisco Redi, John Needham, and Lazzaro Spallanzani. Francisco Redi contributed a lot in the field of microbiology. The ancient belief is that spontaneous generation was first of all challenged by Redi, an Italian physician who carried out a series of experiments on decaying meat and its ability to produce maggots spontaneously. He proved that microorganisms are present in the atmosphere. John Needham was probably the greatest supporter of the theory of spontaneous generation. He proposed that tiny organisms of the animalcules arose spontaneously on his mutton gravy. He covered the flask with a cork as done by Redi and even heated some flasks until microbes appear in the mutton broth. Lazzaro Spallanzani was the other scientist who, by doing his experiments, proves that air carries germs. He was an Italian naturalist who attempted to refute Needham's experiment. He boiled beef broth for a longer period, removed the air from the flask, and then sealed the container. Followed incubation, no growth was observed by him in his flask. He showed that the heated nutrients could still grow animalcules when exposed to air by simply making a small crack in the neck. Spallanzani disproved the doctrine of spontaneous generation. So we are now in the third period, the Golden Era. The Golden Age of microbiology began with the work of Louis Pasteur and Robert Koch, who had their own research institute. During this period, we see the real beginning of microbiology as a discipline of biology. Louis Pasteur is considered as the father of medical microbiology. The concept of spontaneous generation was finally put to rest by the French chemist Louis Pasteur in an inspired set of experiments involving a gooseneck flask. The discoveries of principle of vaccination, microbial fermentation, and pasteurization were given by him. He was the first to discover vaccines for rabies and anthrax. He was the first to propose germ theory. The discovery of chicken cholera vaccine by Louis Pasteur revolutionized the work in infectious diseases that can be considered as the birth of immunology. He gave notion of using weakened form of the disease to provide immunity. He gave the vaccination concept by live attenuated vaccine against rabies. John Tyndall was an English physicist, deal a final flow to spontaneous generation in 1877. He conducted experiments in an aseptically designed box to prove that dust indeed carried the germs. He demonstrated that if no dust was present, sterile broth remained free from microbial growth for indefinite period, even if it was directly exposed to air. He discovered highly resistant bacterial structures, later known as endospore, in the infusion of hay. Prolonged boiling or intermittent heating was necessary to kill these spores to make the infusion completely sterilized, process known as Tyndallization. Lord Joseph Lister. He was a famous English surgeon and known for his notable contribution to the antiseptic treatment for the prevention and cure of wound infection. He is known as the father of antiseptic surgery. Edward Jenner. He was an English physician who was the first to prevent smallpox. He contributed a lot in vaccination. The credit for the discoveries of this first wonder drug, penicillin, in 1929 goes to Sir Alexander Fleming of England. He discovered penicillin from Penicillium notatum. Paul Ehrlich gave his contribution in the treatment of syphilis by arsenic. Ehrlich's work had held important foundation for many of the development to come with the use of salvarsan, marked the beginning of the end of chemotherapy and the use of chemicals that selectively inhibit or kill pathogens without causing damage to the patient. So now we are in the modern era. By the end of 1900s, science microbiology grew up to the adolescent stage and had come to its own as a branch of the more inclusive field of biology. In the later years, the microorganisms were picked up as an ideal tools to study various life processes, and thus an independent discipline of microbiology. Molecular biology was born. The relative simplicity of the microorganisms, their short lifespan and genetic homogeneity provided an authentic simulated model to understand the physiological, biochemical, and genetical intricacies of the living organisms. Many Nobel laureates have their huge contribution in the field of microbiology.